We're going to kick off the afternoon session of Propelling Wonder Stories 3 with a panel that I'm really looking forward to, actually. It's a look at biotechnology, synthetic biology, and the sort of frontiers of very small-scale biological design practices. We've got three great panelists in order from your right. We've got Debbie Chachra, who is a researcher in biological materials at the Franklin W. Olguin College of Engineering. And yeah, that looks better. Who I had the pleasure of being able to write about for Wired magazine, Wired UK's 2010 Year in Ideas issue, focusing on her research, which she'll be describing shortly. David Benjamin is a colleague of mine here at the GSAP, the Columbia School of Architecture, who has a really broad field of interest in architecture and biological systems, and I'm looking forward to hearing what he's working on. And then Carlos Olguin, who's flown out from San Francisco, where he's a researcher at Autodesk. And the description we've been using is very captivating. I've never seen him speak, but it's he works at the intersection of nano biotechnology and 3D visualization, which I'm quite excited to learn more about. So I'm going to throw things over to Debbie, and we'll do a series of short presentations followed by an open Q&A and discussion. So Debbie?
Great, thanks, Jeff. So I'm assuming that there were no larvae in the audience when I chose this picture. So these are sort of figure out how to stand, like not in this way. My background is biological materials. So my undergraduate degree is in engineering, but I mostly study materials that are not synthesized by humans, but are in fact synthesized by nature. So most of my research has been in bone, and I sort of teach about things like, everything from like spider silk to seashells to redwoods. I'm really interested in how the physical properties or the mechanical properties are derived from the underlying structure at sort of everything from the molecular scale on up. So sort of molecular, nano, micro, millimeter scale, right on up to the meter scale. So the thing I'm actually going to talk about today is sort of my sort of fun little side project, which is looking at a type of plastic made by bees. And if you're like most people, and if you're like me about 4 years ago when I started this, you can probably name two types of bees, right? Honeybees and bumblebees. And this is one of the other 10,000 or so species of bees. These are from a family called Callidids, and these are Callidids inequalis. This is a male and a female. This is, in the lifestyle of Callidids inequalis, this is about the last time the male does anything interesting. They live in this particular species. It's a large family. This particular species lives in my backyard, essentially, in New England. And the thing that I'm really interested in is that the females actually dig underground nests, and they do some interesting things. So Jeff, I'm looking at the next slide. So this is a female now, Callidids inequalis, emerging from an underground nest that she's digging. They like digging them in sandy soil. And what they do is they dig tunnels, and the tunnels are about maybe half a meter deep, and then they have side passages that come out. And the side passages serve as nests. So what they do is they provision the nests with nectar and pollen, and that serves as basically a nursery for the egg that gets laid and then turns into a larva. Can I have my next slide? Yeah. So it's a little hard to see. This is one of the nest cells, and it's actually in the palm of my hand, so you can get sort of the scale. It's about a centimeter-ish to 2 centimeters. So it's yellow because it's filled with pollen and nectar. And this one probably has an egg laid in it already, but it's freshly made, so the egg isn't hatched. And the thing that I'm really interested in is that clear cellophane-like material around it. So it turns out, and this was shown in the late 70s, early 80s, that it's chemically a polyester. And so people did what's called mass spectrometry to look at what molecules are present, and they showed that it is really similar to it's chemically similar to the polyesters that we use, and it's not an accident, really, that it looks like cellophane. So it's in the same sort of family of materials as things like polyethylene and polypropylene that we're used to. So we decided to sort of dig this up and sort of learn more about this. And so this is where I sort of have to point out that my background, I'm not a chemist, I work with a chemist, but my background is material science. And I'm really interested in materials, so I'm interested in the physical properties and how that physical property comes from their structure. And this turned out to be kind of useful because it turns out that it's incredibly difficult to find out what this is made out of chemically. And the reason why is because they're incredibly resistant to biodegradation. So these are not, in any sense of the word, compostable, right? They're bio-derived polymers, but they do not degrade. And this kind of makes sense. because the point of this is to be buried in the New England soil for just about a year, and in that process to protect the contents. Because what happens is the egg will hatch into a larva, the larva will eat the pollen and nectar, it will metamorph to Catalyzing bee, and then they'll emerge in the spring and they'll do what I showed in the first slide, and then the cycle will continue. Excuse me. So the point of this is to basically protect the contents from essentially New England weather, which is notoriously bad, right? So it's basically, it can be extremely cold in the wintertime, it can be extremely warm in the summer, it's hot, it's humid, and of course it's buried 1.5 feet below the dirt, which means that there's bacteria, and there's fungi, and there's viruses, and there's also parasites that specifically want to get in there and eat all the food that the Catalyzing qualis have stored. So the purpose of the nest is to protect them against all those things. So it turns out it's really, really hard to study the chemistry of it, because the only way we can do it is by dissolving it overnight in very strong acid at 100 degrees Celsius, which kind of destroys everything, which makes it very hard to figure out the structure. But what we did do is we looked at not the chemical structure, but the physical structure of it. And if I can have my next slide. And what we found, so this is using a technique called confocal microscopy. And what we found was, so there's a few things you can see here. So there's kind of the background green material, which is a plastic matrix, which we know from other people's work is probably a polyester. The little ovoid dots are actually pollen, because of course if it's filled with pollen, we rinse it off and we don't get all of the pollen, so we can see some of the pollen. And then we saw these green fibers. And so the first question that came up is, okay, what are the fibers? And in particular, are the fibers the same thing as the plastic, or are the polymers something different? And what we ended up showing, if I can have my final slide, so this is using the same technique, but this time we've actually stained it using something that is specific to proteins. So that tells us that the red tells us that the fibers are actually made of protein, not polyester. And together with a bunch of other data that we sort of have shown fairly convincingly that the fibers are actually made of silk. So this is unusual for adult insects to make silk, but not unprecedented, which is good. If it was unprecedented, I'd be pretty sure we did something wrong. But larvae, of course, are what we think of as making silk, right? Silkworms, honeybee larvae, in fact, also make silk, so it's sort of known in this family. And so where this leaves us is we have this structure that, if you live in New England, if you live in the Northeast, you've almost certainly walked over these nests if you've been outside. And it's a really sort of humble structure, and it's not very well known. And what we sort of discovered is that there are, in fact, pretty sophisticated silk fiber reinforced composite structures. So think carbon fiber, think fiberglass, right? It's a very similar type of structure that's being synthesized by these bees under the ground. So those are the kinds of things that I'm interested in. So I study biological materials, as I said, I study sort of looking at sort of the phenomenal structures that exist in the natural world and sort of learning what we can from them. So I think I'm going to wrap it up there. You want to? Yeah, we're going to hold off on them.
Yeah.
Go for it.
So we're going to throw things over to David Benjamin. And I'm going to give you a flip. Yeah. You have to get closer. Right? Okay.
Still like Gavin's food.
Yeah.
Yeah, it's been for like 2 years.
It's hard to know.
Okay, so I'm going to talk about a couple of different approaches that I've been investigating, which are basically explorations of the intersection of architecture, and I'm trained as an architect, but the intersection of architecture and computation and synthetic biology. For those of you who aren't familiar with synthetic biology, or even if you are, just to give you a sense of my working definition, synthetic biology is basically a new approach to engineering biological systems or new biological machines to perform useful functions. And so many people think that this has great promise for things like synthetic medicines, synthetic drugs, but I've been also exploring how it might have potential applications for architecture, maybe for architectural materials, but maybe for entirely new ways of thinking about building and designing. So what I'm going to do is briefly describe two projects which are both works in progress. So I'm kind of showing the work kind of halfway through. But also, importantly for me, they're both projects that are more about creating platforms for many possible designs rather than creating a single design for a single scenario. Also both projects involve extensive collaboration, and that's one of my approaches really for being able to take on the complexity of biology and biological systems. So this first one is a collaboration primarily with Fernand Federici, who I think may be here, from the Jim Hasselhoff lab at the University of Cambridge, basically a plant biology lab. We've had extensive work from Jesse Blankenship, who is also here in the audience, so I thank both of them. And basically, these projects begin with a very interesting type of cell called a xylem cell, which occurs in large part in the stems of plants. And here you see a short video clip of Fernand, my collaborator, creating these confocal microscope photos, just like Debbie mentioned, of the xylem cell. So they look something like this. You can translate the photographs through a series of different software applications, bring them into kind of architectural software applications, start visualizing what these cells are like. So that's a single cell and a few different visualizations of that. These cells occur mainly in the stems of plants, as I mentioned. So this particular example, shaped about like this, a kind of cylindrical shape, occurs in the stem of an artichoke plant, and in this kind of naturally occurring formation, its typical formation, it makes shapes about like this. So that red shape is one single cell. But what we wanted to explore is how the cells might operate, how the DNA or the program or the fabrication instructions of the cell might operate if you put it in different conditions. So here we used a great technique that Fernand developed for inducing xylem cells in places where they don't normally exist. In this case, the leaf of an Arabidopsis plant. And so you see the cell executing its pattern in a much more unusual shape, that kind of organic amoeba-like shape. A second approach that we had was to basically grow cells somewhat artificially in a liquid medium through a technique called microfluidics. So this is a plate that you can etch in order to create a custom channel. Zooming in on the channel, you see how we can basically fabricate a bounding envelope or a channel in which the cells are going to grow. The channel is shown in that red shape there. And so that's a shape that we have designed, that humans have designed, and we're looking at the way that a cell will create its formation in that shape. And by extension, we could make many other different shapes. So it starts as a C shape, but we could create an S shape, an L shape, and see how the xylem cell executes its instructions to create that. In this approach, which is kind of a continuation of trying to understand the xylem cell and how it works, we're trying to create a data set from a xylem cell. So we take a few different xylem cells, create a kind of spreadsheet that explains their data, and then we use a fascinating program called Eureka, developed by Hod Lipson at the Computational Synthesis Lab at Cornell. And that program basically takes any data set and tries to derive a mathematical function that explains the data. So we take a data set of xylem cells just in general. Over time, a genetic algorithm runs and tries to compute what is the formula that explains that data. And then once we have a formula, we can take a starting shape like that center rod, that red shape, and execute what is supposed to be the xylem pattern in a mathematical way. So here we're moving entirely into the computer. We start with a straight line and see what it looks like. Then we can use other lines like an L shape and see what it looks like. So we've really been able to abstract the program of what a xylem cell is doing. And in the final potential application, what we're doing is using a layer of genetic algorithms or evolutionary computation on top of the mathematical model to do something, say, at a different scale, which is evolve a xylem skeleton to basically perform an efficient structure, where we set up the evolutionary computation to say, take the xylem skeleton, delete the different bars, and see which ones hold up structurally and use the least material. So really, the whole project was about using the cell as a bioprocessor and basically trying to have the cell help us compute problems at a totally different scale or in different scenarios. So this is the second project, again, trying to create a platform for design. In this case, really thinking about a software program or a software workflow that architects or other non-experts could use to start designing with synthetic biology. You know, synthetic biology, of course, is notoriously complicated and difficult, but as more and more of biology's rules and the rules of DNA become understood, it's possible to encapsulate some of those behaviors in software. And once that is true, then I think it's a good entry point for architects and others to begin using synthetic biology. This project was developed in the Autodesk Idea Studio, partly with advice from Carlos. And basically, the line of experimentation starts with the idea that bacteria already create incredible spatial patterns, shown here in some photographs, some by Fernand, some by others. And it's also possible, as of recently, to start to encapsulate that behavior in software. So bacteria already create these patterns, but you can also have software that allows you to change the pattern, and change the inputs or the parameters and change the patterns. And this allows us to basically translate from the scale of single cells all the way up to patterns that we can see and touch, and that are at a scale that we could start thinking about them as materials, even building materials or architectural materials. So you see there the last one is about 1 centimeter in width. And so this is the part that we built over the summer and that we're using with students at Columbia, which is basically a way to allow a series of input parameters to be adjusted. So you can adjust a kind of bounding box to execute an experiment in. You can create a lattice of, say, 3D printed material, that's the gray stuff, and then you can basically execute an experiment in the software which will simulate the way that bacteria will interact with one another. So here you see red spheres and green spheres colliding with each other and sticking to each other and avoiding each other, and that's basically a simulation of the way that two different types of bacteria would interact when you program them to do things like emit a chemical or seek a chemical. And so we can do that in a certain software program, basically building functionality into Autodesk Maya. And then once we can generate a pattern, then we can kind of mesh that geometry, make it into a solid geometry, and use a different piece of software, again developed by the same lab at Cornell, Todd Lipson's lab, which will allow us to say that if we have one solid object made of two different materials, red material and green material, how will it perform as a single structural object? And this is a simulation of that. So you see different patterns will cause different structural performance, different stress levels, and then we can use, again, a kind of automated computation process to evolve better and better designs over time. How might this be useful? What we think is that it would allow us to create a single object, like this flat surface, with different regions based on bacterial patterning, the red and green, and it will perform in very specific ways according to very specific and very complex patterns, but patterns that we can start to control and identify. The potential larger application of that is if architecture long ago combined the building's structure and skin in the same object, like think of a stone-made building or a brick building. Modernism was about a time when in architecture the skin and the structure were separated. Think steel structure and like a glass envelope. And now we may be back at a moment where we can think of biology helping us to create a single object with structure and skin again integrated, but using biological machines and genetically engineered machines in order to create these very complex patterns that could create new building materials and potentially new types of buildings. Thank you.
Just briefly for people who've walked in, if you're looking for a chair, there's kind of like islands of chairs hidden around the space. If we want to carve and let people walk in so that they can sit down. There's a couple chairs up here, there's one up here, etc. So feel free to make yourself at home. Yeah, if we
It's
can.
probably easy to, yeah.
It's probably easy to maybe if you can sneak around these guys and you can come into the middle. There's a couple chairs. Sorry, thanks.
Okay, hi, I'm Carlos. I work for Autolist Research in San Francisco, and I'm really excited to share some of the things we've been doing in collaboration with other people like David. First, maybe some of you know what Autolist is. We traditionally focus in industries like AEC and manufacturing, and here are some examples. So you can see at the top computational fluid dynamics simulation in a building, the envelope of the building. You can see also examples of solar incidence radiation into the inside of a building and trying to understand correlations between, let's say, the size of the window against the heat power needed to warm the building. And examples on the right are about car industry and manufacturing, and on the left is more about visualization and going into games. I think for the last 18 years, every nominee for an Oscar award in computer graphics and special effects has been using our software. So it's kind of cool, and at the same time, all of that is dead. So we're interested in what is the living space as a design substrate. Here's another example quickly also about reality capture. So it's not necessarily novel in some ways, but basically it allows anybody with an iPhone to take, or any smartphone, any camera, to take lots of images and then create a 3D model. It's a full 3D mesh. And you can start to imagine the things that can happen out of that context. Again, what is more interesting for us is to think about the applications. If you can suddenly scan something that you like, modify it, and then you print it out, that's a world that many think we will live in not too far from now, and it resembles some of the things that have been going on right now. You can apply that same technology to scan a building, and some of the people at Autolist are putting cameras in quadcopters and being able to take these images out of buildings and be able to get a model out of the building, and then be able to modify it and then create some extensions to it. You can apply it to humans as well, and this is where we start to connect to life sciences. This is Robert H., by the way, for those architects in this room. And people have discovered moles that they didn't know they have. But it's again interesting to think what you do. So again, the main point of these three slides is there is a pattern that we relish at Autolist, which is scan, modify, print, or capture, design, and create. And basically, this is the scan part of it. Now, when we think about life sciences as a space to explore, first you can see on the left all the industries that we support today. And then you see on the right life sciences, and you see some of the disciplines that we use in these other industries, which is visualization, simulation, design, analysis, etc. So what I'm going to do in the next 7 minutes or less is just do a quick rundown of some examples of what's going on in life sciences that we're part of so far. I'll start with visualization. It's the most obvious entry point for us. And of course, this is work done at HeartWorks. this is an animation, it's not a simulation of a heart, but it gives you an example of how people in science started to use software that was normally used in films and games to be able to illustrate scientific phenomena. Going more into the molecular level, this is work done at Harvard and designed by Campbell Strong and Gil McGill, and this is a ligand binding to a protein. And it's halfway between an animation and a simulation. There's thermal motion going on in the atoms, and that is really a simulated behavior, but how the ligand binds to the protein is not necessarily so. But it's interesting for us because as the animations become more and more driven by algorithms, you can imagine that suddenly starts to feel and look like a simulation. We, as part of this effort to explore, we're just starting this initiative. We worked really hard to help people in competitions like iGEM, which is an international genetically engineered machines competition, which is really a competition around synthetic biology, to help them. These are all undergrad students that go to MIT every year and try to produce interesting projects around synthetic biology. And we try to enable them with software that can help them visualize what they're doing. You can take one step further, just a very quick parenthesis. You can also visualize by printing it out. And I don't know if Paul Gibson is here, but we're very inspired by his work. So this is an actual molecule printed 10 million to 1 scale, and it's huge, and it looks almost like the face hugger in Alien. But it's a ribosome. So it's only the top one part of the ribosome. It's the 50S unit, and you can see here already finished. It still has a support material, which we thought it actually looks very cool with the support material. We're going to remove it soon. This is just from 2 days ago. And it again gives you another perspective when you see things this big, and there are scientists that have been working for decades with ribosomes, and suddenly seeing them at this scale gives new perspectives. So that's interesting in itself. George Church, who we collaborated with to do this, he was telling me he's holding a cup, and he says, look, the guy who designed this cup has this constant feedback when he's designing it. People working at the molecular level don't have that feedback. It's all based in directions and statistical information, equations, etc. So anything we can do in that sense, it's helpful. It's just one more, another view of it. Okay, so let me show now a quick example about simulation. So again, now moving into simulation, this is very basic. All we did here was that we took a paper that showed a diblock copolymer, the experiment results of how the microstates of diblock copolymers are shown based on the concentrations of one material against the other. And the only novel thing, I guess, there is that we use Maya to see if Maya would be able to reproduce qualitatively those results. And we did, so we were very happy. So again, we're not trying to claim there is quantitative data that can show or validate what's going on, but qualitatively it was able to show the same patterns. And you can see that based on the concentrations, you have these different patterns, which evokes a bit of some of the work at a higher level at the mesoscale David was showing for E. coli, for bacteria. What's important is that if you change parameters at this scale, you change variables at the macroscale, like if it's more stiff or elastic or things like that. It's also interesting, and I'm going to just jump here to show a quick demo how this could actually be played with in some ways. So I'm going to, so this is Maya. So this took me about 10 minutes to do. So the purpose is not to show that it's novel. In fact, the work that David showed before Maya is far more complex. It's just to show that it's easy to do at a certain level. So these particles, this is my computer, my Maya here running on my computer. These particles there are two forces in tension. It's very basic. The green parts are attached to the red parts on these pairs, and then every green part attracts each other following a so-called van der Waals force. Okay, and every red part attracts each other in a similar way. So with those two forces in tension, anybody want to guess what's going to happen when I play the simulation? Okay, I'll play it. So it's a little bit similar to what I showed before, where you have these lamellar conformations of red stripes with green. You can tilt it so you can see a little bit more. That's pretty fast, so maybe it's not easy to see. But it's interesting, just with those two forces in tension, I can get this pattern. So I can, for example, now turn off one of the forces, like say the red parts that attract each other, I'm going to turn them off. Anybody wants to be brave enough and try to guess what would happen? What kind of pattern might emerge? Okay, so I'll turn it off. And now I get, the red parts are not anymore attracting each other, so it just hang around the green parts. And even at this basic level, you may even just wonder, so why that size, or why not a big lump, or for us it's motivating to be able to help people at all ages, for that case. I mean, children, youth, and researchers, to illustrate the types of things that they're doing, and slowly maybe start to get into simulation in these qualitative ways. If you were wondering why is it in a plane, that's mostly because all the forces that we apply, this is just an exchange between kinetic and potential energy in this constant state. And if I wanted to be completely accurate, I could, for instance, calculate the total energy on every time step and make sure that it's always the same, and things like that. I can go as accurate as I want. But just for the purpose of one more quick experiment, I'm just gonna move the red parts up or maybe down. So, and by doing that, now even though everything was in a plane before, it would be creating a momentum that then the forces start to go in the z-axis as well. So again, this is just a 10-minute brainstorming. So I'll just move quickly to show other pieces of work. And so design. Okay, so what we're doing about design? So this is obviously nanite, and yet that's at the nanoscale. And this is the work done by Sean Douglas at the Wyss Institute at Harvard and others, also within the George Church lab. And this is DNA. So DNA is being used as a structural component. DNA is highly predictable in the way it folds as opposed to a protein, for example. So using the basic rules of how DNA folds, one can create this so-called DNA origami. Now you can imagine this is the perfect fit for a software company to try to create a software tool that enables somebody to then create more easily these shapes and let all these calculations of how the different nucleotide bases pair to each other be abstracted from the designer. So this is an electron microscope image of that same triangle being folded, and per the design, in some cases it didn't really fold, but it gives you some flavor of that. This is something we showed at TED Long Beach earlier this year. So we actually brought an electron microscope, a desktop one, so that's kind of novel too, and you can see collocated the image next to the design. So here are more examples of the work done at Harvard. And again, the modeling line on the left and the images from the TEM on the right. And here's more examples of the things that they have created. It's amazing the flexibility of the shapes that can be created. Again, without software, this would take weeks or even months to do. So we were working with them to help accelerate that path. Now, you might also wonder, I alluded briefly about how to abstract that. So here's one example. This is a prototype where the user really, for that case, doesn't need to know anything about biology. It's just creating a volumes distraction between two boxes and creating this nutshell. And so you do need to know 3D modeling software like Maya, but as he pushes a button, it gets converted into DNA. So in other words, the 3D shape is converted to a DNA-based shape from which you can then output the right series of sequences that then anybody can order to something like GeneArt or any DNA synthesis company, get them back, put them at room temperature, and together, just by being in the same place, they will bind naturally into the most favorable position, which would be this type of construct in this particular case, all defined by the code. So again, we're using, or they are using DNA as a structural element, not to create proteins, not as a blueprint for proteins. There's a competition just happening next week at Harvard. It's called Biomock, and this is the first time that they're going to do this. And about 22 universities from all over the world are going to come and try to do things. About half of them would be related around this technology that I just showed you. So it's something to and might need to follow, and we're happy we're part of it. Let me show you one last demo. So this is an extraction of DNA origami. This was done by Yo Stam. He won an Oscar award in computer graphics at Autodesk. I think it's not called Oscar, but it's given by the same academy. And it's he decided to start to explore this space, so he read some of the papers, and then he took some of the properties of how DNA folds and put them into Maya. So I'll just play the video really, really quick. And it starts by very simple shapes. So again, now we are not just enabling somebody to design something, but you can actually start to simulate how would it fold into a certain shape. And that actually becomes a little bit more interesting down the road, because as you imagine, this technology, which is at the nanoscale, and by being at the nanoscale, it means that it can interface, in theory at least, with other nanoscale technologies like cell biology. Then we can potentially interface with biological systems in the future, and this is exactly what Harvard is doing. So for us to start working on things that enable the simulation of these parts, it's interesting to the point that at some point you might just give a certain input, let's say a protein, and you want to say, I want to create a shape that binds to a certain site in this protein in a certain way, or attaches it in some way into a certain place. DNA is probably too big to bind to a certain spot in a protein, but it can position it in a certain way. This is for TED. We just as a proof of concept, we did this experiment also. And again, it's all bottom up. Okay, so it's all self-assembly. There's no top-down approach. All these individual parts have constraints from which, in interaction with others, create these emerging shapes. What is also interesting for me to notice is that we didn't learn this type of design paradigms when we were kids. We learned for the most part this as we were adults, self-assembly. We're used to cutting and slicing things with our hands. So I'm curious about what the new generation of scientists and designers, as they get exposed to phenomena like this early on, what they would do, in 20 years from now, 30 years from now, when that just becomes so natural. Am I doing okay on time?
Yeah, we should just wrap up.
Yeah.
Okay, yeah. So I'll just finish one last project, which is about analysis. And this is, so this is called a bioweather map project. It's done with the Personal Genome Project in UC Boulder, the Knight Lab. And let's see. Something's going to be wrong. The resolution there. Okay. So what we did here is that what they have is an initiative where they are capturing DNA, bacterial DNA on currency, that then by the fact of being able to capture the DNA on the currency from different parts of the world, from different capture points, they can start to eventually to create this notion of a bioweather map, where you see the bacterial DNA moving from place to place, mutating, etc. And in this particular conceptual, movie. It's illustrating how a potential outbreak could be identified early on. But again, what it is done right now is that we're capturing the currency. So this again was shown at TED Global, this time Edinburgh, and we had this little ballot where people would put their currency in an envelope, and you can see some of the examples here. So this is a bill from Malaysia, and it has here all this is a phylogenetic tree over here, so you can see here the bacterial DNA. These are OTUs that represent the bacteria in this bill. And we also did, this is a demo app, really, it's not a real app in many ways. We show some of the bacterial shown in the bill that has as a natural habitat human beings. So in this case it was Priotella, so that's normally lives in our gums, in other places in our body. And this is another bill. And again, it has a different conformation, genetic conformation. This is yet one more. And again, we try to illustrate where it would normally live. This is the total number of bills so far captured at that time. And you can't see it by country, but the resolution is not high enough. But it's I'll finish just by saying it was a way for us and the researchers to raise awareness about the fact that, first of all, we use bacteria, so bacterial currency is safer than trying to get DNA from humans, especially in the European countries, which are more protective, perhaps, than even ourselves. But it's a way also to say get used to it, right? I mean, today's currency, next time is going to be you. The fact that you think that your DNA is something that you want to keep private is as if you were saying that you want to keep your face private. We just happen to live in a snapshot in time where we don't know enough about the traces around you to be able to capture that DNA, but it's going to be very pervasive, and it's probably easier to just get used to that and learn how to live with those new dynamics. So anyway, I'll stop here and thank you.
Cool, thanks, Carlos. That's a slightly ominous end for the presentation, actually, but that was fascinating. So we're just going to throw this open for a brief panel conversation amongst the three of you, and I'll just start off with one question, but anyone in the audience that wants to chime in, feel free to, and we can either get a microphone to you, or you can just, I'm sure we'll hear you. I'd love to just start off with pointing out just sort of two different directions that sort of come out of the work. And I suppose that this question is really for Debbie first. There seem to be at least two directions. One is extrapolating from a natural phenomenon certain material. That might catch on fire. Or we can try to put the filler on. Material, spatial, or other kind of chemical lessons from a natural phenomenon, and then turning that into an artificial product later, whether it's a pharmaceutical or a building material. And then the other direction, and this is maybe more in what David was showing, there's the idea of taking artificial technologies such as 3D printers or other kinds of genetic material, and actually then generating or constructing a quote-unquote natural object, kind of augmented nature or augmented ecology in Simone Ferracina's sense. So in any case, I'm curious about just that idea that there's either learning from bee plastic, so that maybe in 5 years, I'm going to be wearing a Swatch watch that's made out of bee plastic or bee polyester, and then the opposite, which would be, I guess, a question for David afterward. But so could you just talk about that idea of extrapolating from natural phenomena?
Sure. I mean, there's sort of two. I mean, so there are an enormous number of incredibly complicated structures that already exist in nature. And there's, of course, an infinity of possible things that we can develop as humans, right, as a species. And so I think it's not, I certainly don't feel like that's a dichotomy. I certainly feel like it's learning one and using it to inform the other. And then having said that, in the context of what we're talking about today, there's the other piece of it is the idea of the sort of the structured material of the body. So, for example, Carlos was talking about making these structures out of DNA. And of course, in nature, structures are not made out of DNA, right? I mean, so actually I love the idea of structural bionanotechnology, because what I actually teach and do research on is structural biomaterials. So there's a set of materials that are actually used in nature to make things, including cellulose, including collagen and elastin. There's sort of a well-known sort of subset of typically proteins, but also ceramics and other things that are used to be structural. So the building blocks there are a very different set than the set of potential building blocks, which is things like DNA, but also things like, so synthetic biology, there's 22 amino acids that everything is made from in biology, but it can code for about 64 would be the theoretical limit, but it can code for a couple dozen more, probably. So the tension piece is both what it's actually made out of, but so thinking about what are the building blocks, and then thinking about what kind of structures we could make if we had a different set of building blocks. So I'm going to turn this back over to you to talk a little bit more about the structures.
Yeah, well, I guess I agree. I don't see it so much as a dichotomy, especially because from my point of view, I'm looking for ways that, as a designer, I can use any type of new materials, new technologies, expand my palette of possibilities. But I think it is an exciting time for me to be talking to synthetic biologists, because potentially synthetic biology offers an explosion in design possibilities, like a huge leap in what is possible besides mining nature for existing. Although clearly there's like a lot still to be mined for nature, right? I guess one of the things that's become interesting to me in trying to work with biological systems, specifically synthetic biology and synthetic biological systems, is how we, first of all, see those systems, how we can design something that we can't see, but also how, as architects, if we always have to incorporate a lot of design requirements, the typical ones in a normal building would be like structure, program, circulation, the site, aesthetics. I think now, like, there's this kind of huge increase in combinatorial possibilities when we also talk about new types of materials and many new things to consider. So one thing, for example, if we go back to structure, like thinking about structure at the same time we think about other design features of a building or building material, I think that is super interesting. So not only how can we design new types of structures, but then how do we integrate that with all of the other requirements of architecture or building material. But maybe more specifically, something that has always been on my mind as I've been working with biology in the past couple of years is that it's a very strange world and probably just newly possible to be designing at such vastly different scales simultaneously. In other words, people who are already using synthetic biology to make biofuels are designing simultaneously at the scale of DNA, which is like the radius of about 1/billionth of a meter, and at the scale of climate change and the Earth with a circumference of 40,000 meters. And they're thinking about like the same product and the same system at those multiple scales simultaneously. And it's really fascinating. The same, I think, would be true if one were to genetically engineer bacteria to produce new building materials, which might be able to be produced with sugar instead of with petroleum, and you would alter global trade and maybe carbon emissions. And I think there are just some really fascinating possibilities ahead. And then how do those whole ecosystems intersect with the actual product of a single building or a new building material industry?
So, and of course, that's one of the things that is actually handled extremely well in the world of nature, right? Just think about the scale of coral reefs that are made with polyps that are sort of on the sort of millimeter to micron scale, or think about your xylem cells, right? Those are the building blocks of redwood trees, which are on the scale of meters to hundreds of meters to. Does anyone know what the maximum height of a redwood tree is? It's definitely in the hundreds of meters, right?
400 feet.
Yeah, so like in that ballpark. So these types of things are addressed pretty routinely, is probably not the right word, but sort of are addressed in nature. And that's one of the things to me that's really remarkable about biological materials, that their structural properties at the macroscopic level are really encoded in the molecular level, right? So, I have a favorite protein. Of course, I have a favorite protein. It's collagen. And collagen is a rope at a molecular level, right? That basically, it's literally 3 strands of protein that are twisted together. And collagen makes up, it's in our skin and our bones and our ligaments. So at sort of and our body scale is sort of on a centimeter scale, and it is essentially engineered from the bio-nanotechnological level, so down at that level, to have the physical properties it does. And the materials that we currently use to make buildings kind of aren't, right? So I feel like that would be transformative to architecture.
Yeah, I think that certainly seems true, that there could be whole new ecosystems of the way that we create materials, and that would change the way that we design, also the way we construct on site, and what new designs are possible, what a building is might change as well. Just to offer one other perspective that I think ties into everything that Carlos is doing, is that I found that even though my interest initially was exploring this exact kind of thing, what new types of buildings might be possible from new technologies of synthetic biology, something like that. Immediately, as I started to explore these and try to develop tools or think about what new things would be possible, I started thinking about and using software. Now, partly that was probably because that was something that was already familiar to me, but it's a hypothesis that I'm developing now. I'm kind of just crystallizing something that I've been thinking about for a long time, so I'm not sure if if this is a well-tested hypothesis, but the hypothesis would be that it wouldn't be possible to explore this stuff without software. I mean, the software is so key that even if we're just talking about buildings and building material and natural systems, the computation is so essential to it that we need that aspect to be an integral part of the brainstorming unit at the beginning and all of the possible solutions and the features and the way that we're going to do this.
I was really taken by Carlos's comment that how you design differently if your root tools were the tools of computational synthetic biology or computational biomaterials. I actually want to turn this over and ask you about that piece of it, right? So we're talking about new materials, you're talking about how do we have structures with new material, and as soon as we talked a bunch about, okay, well, how would you actually do it? What would be the tools?
Yeah, so I agree with your point, and maybe I would also add that at the same time, software is this training event, right? Somebody designed that software based on some user activity that, if we were lucky, he actually followed a user to be able to create the software. And most likely in the industry in general, he came up or she came up with that grandiose idea of how the software should look. And then everybody's constrained to follow that process. So that's the counterpoint. But at the same time, I recognize, without software, it's like a brain prosthetic that enables you to do things that you couldn't otherwise do just by the massive scale of computation that is doing for you in seconds. So I'm almost curious, and we were just talking about this, David and Ivy, earlier today, when we're looking at this space, and this comes to your question, we want to come in with both an open mind of what is this new design space affording, and what are the activities that we can reflect through software. But at the same time, we want to say there are design patterns that can be repurposed from all these other industries, design spaces that we can maybe learn from as we approach a new space. And I don't know the balance, but I know it's somewhere that it can be one extreme or the other.
I would like the maxim that we shape our tools and thereafter our tools shape us.
Yeah. Are there any questions in the room for the panelists?
So I was really fascinated by the ribocyte, the 3D ribocyte printout, and the way that students can look at a fundamental building block very differently by being able to literally look at it. And then also your slides, being able to stain the protein and see it there and compare it to other things. Are there more areas of visualization and physical interaction that you see that are changing the ways that synthetic biology is accessible to designers?
knew about Fluidics, so there's a group at the University of Washington that is looking at protein folding problems. So it turns out, so an interesting thing, and I think there's actually a huge sort of opening for this, which is that the way that proteins interact with each other is basically geometric and physical. Like it's a tactile interaction. It's a tactile interaction. So you showed the ligand. So it's a combination of forces that you have sort of a 3D thing that fits into another 3D thing, but it's not as simple as it's not just a job. It's not like a lock and a key, right? There's actually an interaction between the forces, similar to what you showed in the Mayo demo with the two components that actually interact in different ways. So think lock and key combined with that Maya demo where you have interacting forces. So the big frontier, I think, is actually in creating sort of tactile models that incorporate both of those two things. So where this brings us to Fluidics is a program that's created at the University of Washington, and it's a game. It's actually a game to try to fold proteins so that they not interact with other proteins, but actually take the proper three-dimensional configuration. And it turns out that humans are a lot better at seeing that 3D configuration that they can take, subject to all the sort of interactions of the different parts, than we are computationally. So it's a hard, it's not an intractable problem, but it's a hard problem computationally. But humans are actually really good at just looking at these you saw the ribosome, right? Super complicated three-dimensional proteins, and like moving them around until they actually get a shape that's a plausible shape for them to have. So yes, right? Because it turns out that the nature of how proteins interact is so physical and so tactile, but at a molecular scale, that it seems like there should be an enormous amount of room to do things that are both physical and tactile for sort of how we can think about how to interact with them or how to understand them. I'm not sure if anyone's making, now I want 3D models that have like implanted magnets so that they can interact. Do either of you know what you want to add?
I know there's somebody, Art Olson at the Scripps Institute. He has, if you Google him, you'll see an augmented reality demo of a molecule, and he's moving around. And then he has another example where he uses magnets. And he has this virus, and all the different parts have magnets, and all he does is just put them in a glass container and shakes them. And just by the shaking, it emulates this thermal motion environment, and eventually they assemble similar to that video, but in reality. And I think that's also very key. I mean, I would say they're more like, at this point in time, I wouldn't say toys at all, but they're not yet used scientifically, but eventually they might be used.
So the Fluidics actually, just a couple of weeks ago actually, they announced a scientific paper it was for a protein involved in HIV, and the structure was based on something that came out of the game. Someone on the leaderboard basically came up with that design, and they obviously did further studies. But so it's certainly starting to have this sort of physical models of molecular things is actually starting to have an impact even in scientific research.
And I'll just add that there's an incredible computer scientist at Columbia, actually, named Eitan Greenspan. He's developing some really incredible tools, not that have anything to do with biology, but that have to do with the relationship between being able to visualize something and being able to design something. So normally when people say design a car and they want to test how aerodynamic it is, they have to send it out for analysis. So the CFD testing takes several hours. It's often done by specialists. And something that Eitan and others are working on is being able to see real-time simulation of changing the shape of the car, and you see immediately how it's more or less aerodynamic, and many complex versions of that on one side or the other, crosswind versus drag. And I think that is going to be a really exciting frontier for biology when there's enough known about the biological forces to have simulation that you can experiment with in real time. And Carlos's example is, I think, a great way to think about the way that's going to work. Some biologists are starting to develop custom tools, custom software tools, and many biologists that I have come to know have biology labs have computer scientists in their lab to develop these custom tools so that they can say, one example is something that I've used, change the different number of bacteria, 2 different types of bacteria interacting to see the different pattern, or change, in theory, the DNA of that bacteria and make it seek a chemical more or less, and then change the pattern, and you can see that stuff in real time. And I think that's going to be a whole new frontier.
I can add, just one quick comment to that is that I think there's also a trend to coarsen our models so that they give enough information to what you actually want to do. Because obviously, if you want to be completely accurate, then we don't have yet that computational power, or even the way to model it in full. So there's a trend to be able to simplify as much as we can and see if that yields the types of representations that you're looking to work with.
And actually, before the panel, David and I were talking about, there's a company, I believe it's called Nervous System, that makes 3D printed jewelry that's based on basically biocomputational patterns. So it looks at patterns in nature, it figures out how to replicate them in software, and then makes really beautiful, sort of instantly organic looking jewelry out of this. And that's a good example of abstracting out all of the biology and abstracting it out into patterns that you can actually then sort of do interesting things with.
Let's just take one more question. Is there anything in the room? Jen?
I guess my comments are mainly a compliment, but as a historian of science, I think of different sciences that became computational at different times. And it's relatively easy to calculate an orbit with your simple rules. As an astronomer, they were doing it in the 19th century, even before that. And then in meteorology, which I talked about, the 1940s really made a big difference. And there was this progress from understanding to prediction to control that went pretty quickly. And certainly an architect would have to predict the outcome of your materials and try to control the qualities of your building. But in meteorology, they thought that that would lead to weather control. I didn't talk about it in the talk, but there's a sense of a perfect forecast could allow you to intervene at any point to get out in the field and make an intervention in nature. So they were almost thinking of architectural meteorology, I guess I would call it. It's kind of a new neologism for me right now. And I think the same thing's happening in climate. Seeing the models are extremely complex, but partly predictive rather than just sensitivity studies. And so computational biology, I think it's an exciting, and computational architecture with materials is a new kind of exciting kid on the block, but the history of it has something to offer, which I said it was more of a complementary reflection. The question is that I'm also a kind of a buddy of, our recently departed Ed Lorenz with his chaos, and we're certainly dealing with some fractal properties. Do you have any computations that kind of blow up, or they don't form the nice little buckyballs, or there's a chaotic element that comes in as they discovered, in about the 1960s with their equations, their nonlinear equations back then?
Okay, well, the only example that can come up is that in the film industry, that's exactly the original goal. Like, you create these explosions, they are chaotic, and using the simulation engines, the same ones that now we're trying to use to create order by changing the constraints. So it's interesting because thinking of your talk about history, I do remember reading some papers from 10, 15 years ago where people in the film industry were trying to figure out if they could, by exploding something, the easiest way would be by making it, they were trying to follow patterns of how to come together to a self-assembled component. And was the way to learn about how something self-assembled would be by exploding it and then trying to reverse it back. But that's the only reference I know.
So I was just sitting here looking thoughtful, trying to think of any examples of chaos in biology, and I'm going to take this as a challenge now, like go and do some research, because I literally cannot think of a single example. The one thing I can think of, though, is Kevin Kelly, I think, is the person who coined the term gnarl, like a gnarled tree, and he basically used it to describe sort of chaotic patterns in nature. And so the thing I think about is, like, if anyone has pets, like sort of the sort of pattern of fur around their sort of face, like where which direction the hairs go in. Think about tree roots, the way tree roots are sort of bent. Think about the way, if you look at a meadow, think about the way that the grass is bent in position. And so it's not random, but it's also not patterned. And so he used the word gnarl to describe this, and it turns out to be, I find, a remarkably useful term, that sort of characteristically natural-looking randomness. But I find a hard time thinking of things that are truly chaotic. It seems like almost everything has a negative feedback loop, so it actually tends towards the sort of structure that makes sense rather than sort of blowing up and being chaotic.
I was told once that slight asymmetries in your body, one leg's a little shorter, that gives you a feedback that allows your balance to actually stay on. If you're perfectly symmetrical, you might just fall over.
Right.
And so when you're moving, you get slight little, it's like jet control mechanisms. When the jet has a little turbulence, that feeds back into the instrumentation and keeps it steady.
Yeah, so the thing I think about is, so I spent a lot of time studying bones, so it's one of my sort of go-to sort of mental models. So your long bones are sort of more or less, they're complicated, but they sort of look solid. And then at the ends of your bones, like in your hips, it's, think about a sponge, right? It's like a three-dimensional pattern. And if you take a slice of it and you look at it, it looks really random, right? It just looks like a sort of a three-dimensional random sponge. But it's in fact heavily controlled by the stresses that go through it, something called Wolff's law. And if you have lots of bone there, it kind of looks random, but as you age and you lose bone preferentially at places that are not on the stress lines. So you can actually sort of see where the stresses kind of emerge from this random structure. So that, to me, is sort of a good example of something that is apparently random in biology, but it's highly epigenetic. It's highly governed by the sort of patterns that are embedded in it, even if they're not apparent when you see them. But that's an excellent question. Chaos in biology.
And maybe I'll just offer one comment about kind of the first half of what you're observing. I'm super interested in examples from history of similar types of things, because while I'm fascinated by the new possibilities, I'm also aware that it's not a completely new framework, and that we might draw some interesting lessons from things like, so now I'm going to look up that chapter in your book. Is that a chapter in your book?
Which one?
Well, the one about the meteorology in the 40s.
There's a 1945 report on perfect weather.
Yeah. But it made me reflect just now that one, a lot of the biologists I talk to say they're very clear that we're not quite at the level yet where we can use computational models to accurately predict things. So they're aware of their own limitations, or the limitations of the science behind it. But kind of as a complementary thought, one thing that many labs that I've talked to are starting to do more of is use computational models in tandem with physical experiments. And that feedback loop, use the computational models to explore wide design space, lots of different possibilities, very exploratory, maybe not super accurate, maybe it's coarse models like you're saying, Carlos. But there's, it's easy to do that quickly, not much lost if it's wrong. And then some of the best hypotheses you test in the actual lab. And it strikes me that maybe something different about that and weather control is that you can actually do the experiment in the lab, whereas it's much more difficult to experiment with the weather. I think people try it,
We're doing it now outside.
Yeah, but I think that's a really interesting trend in the near future for the exploration of say design with biology, which would be heavily informed by computation, but also tested or verified by the lab, and that would have to be in a kind of constant iterative loop. And that the way, say, an architecture school works if you need that lab experiment.
Can I say a second comment? Reminding me of evoking your own work, the other third component would be, in my view, biology is computation as well. So if you can harness biology as this massively parallel computational power, that is an design element to introduce into the equation of how to solve all these intricate problems that we have.
Cool, thanks guys, that was great.
Thank you.